Hello everyone, and welcome to this audio podcast. This audio podcast is part of Quail and Crux. This audio podcast is part of the Ricochet and Ruckus. This audio podcast is part of the Shitstorm Squadron. This audio podcast is part of the Bedlam and Labostix. This audio podcast as part of a ZtormLabostix production. So this in this audio podcast, this will be the sort of final audio podcast in this series, in as part of and this audio podcast will look at the conclusion, some of the conclusion in around an overview of Huntington's disease. So recent development of of and through analysis of what's referred to as CRISPR and in around CRISPR and specifically and specifically in around Cas9 or CAS9 gene editing technology or analyses brings a sense of hope to those who suffer with Huntington's disease. So while still in its early stages or phases, rodent models or investigations have informed sort of helpful or useful in uncovering sort of the more effective pharmacotherapeutics, pathogenesis, and biomarker development of Huntington's. Murine or mice with heterozygous Huntington's and knock in human Huntington's on exon 1 have been treated with CRISPR or through the use of CRISPR Cas9 system aimed at decreasing the expression of mutant Huntington's in a striatal neurons. It has been theorized at the same time that part of the progression of Huntington's is sort of a decrease in arteriolar cerebral blood volume or CBV, and therefore at the same time inflow based vascular space occupancy and MRI imaging has been documented or evidence in works from publication from 2021 by investigators to determine the effectiveness of CRISPR Cas9 on the angiogenesis in the in particular sort of regions of murine or mice investigations. The team actually there's one team in amongst their works from 2021. They focused on two parts of the brain, the motor cortex and the striatum, and mice were subjected to several different behavioral tests or investigations to assess the efficacy of the gene therapy. The mice were injected directly into the striatum. The mice were injected directly into the striatum with radio labeled viral vectors and the in amongst the literature. Additionally, rodent investigations have yielded sort of promising sort of analyses as in the same or similar to mice or murine investigations investigations and where throughout sort of the public throughout where investigators have sort of have sort of in, have sort of delved into the analysis, delved into the analysis of Huntington's and Huntington's disease. Radio labeled analyses has been sort of one of the main investigate sort of techniques or ways to determine the sort of neuro complexities, the neural complexities and deficits in around Huntington's. So that draws this audio podcast to a close. This audio podcast is part of the Quail N Crux. This audio podcast is part of the Ricochet and Ruckus. This audio podcast is part of the Shitstorm Squadron. This audio podcast is part of the Bedlam and Labostix, and this audio podcast as part of a ZtormLabostix production.